Hello everyone, I'm Nicoletta Rocca, Associate Professor at the Department of Biology at the University of Padova in Italy, where I teach plant ecophysiology and remote sensing of vegetation. My research has long focused on how plant and photosynthetic microorganisms respond to environmental stress and also on the characterization of cyanobacteria and microalgae from extreme environments. And about 10 years ago, this work led me into the field of astrobiology, where I joined a multidisciplinary team aiming at investigating whether oxygenic photosynthesis could occur or could be feasible under simulated exoplanetary conditions. This is also the topic of the EAI seminar beginning on March 24, that is titled Photosynthesis under Simulated Red Dwarf Spectra and Analogous Light Niches on Earth. The oxygen photosynthesis is particularly interesting for astrobiology because its evolution completely transformed the Earth's atmosphere and made our planet habitable. It also produces atmospheric biosignature linked to the oxygen and surface biosignature as related to photosynthetic pigments, making it a key target for the search of life beyond Earth. However, many potentially habitable exoplanets orbit red dwarf stars that are quite different from our Sun. This type of stars emit in the very low indivisible, while are enriched in infrared and near-infrared radiation, whereas the Sun spectrum is dominated by visible light. This challenges the photosynthetic process as most oxygenic phototrophs rely on visible light to drive photosynthesis, and differently, infrared and near-infrared light are less energetic and generally do not support conventional oxygenic photosynthesis. So a key question in astrobiology is whether oxygenic photosynthesis will function under red dwarf spectral conditions and produce a detectable biosignature. To address this, we studied the diversity of photosynthetic microorganisms and organisms, and we performed laboratory simulation. During the seminar, we will show you how we study the biodiversity of the photosynthetic organism and to better understand the astrobiological implication of photosynthetic microorganisms that are able to utilize also red light for photosynthesizing. And I will also introduce our laboratory platform to simulate key exoplanetary conditions, including anoxic atmospheres and different stellar spectra, along with the real-time monitoring of oxygen production by the phototrophs when exposed under non-terrestrial conditions. Finally, I discussed the experiment we carried out so far and the result obtained to help to define the limits of oxygenic photosynthesis, its feasibility on planets orbiting red dwarf stars, and what kind of biosignature it may produce. I hope to see many of you next week, and thank you in advance.